Welcome back to the Deep Dive. It is Wednesday, January 21st, 2026. And I have to say, looking at the mountain of research and data we have pulled together for today, the vibe is, it's just noticeably different.
Different is an understatement. I mean, if you were tracking the biotech sector back in, say, 2023 or 2024, it was grim.
Oh, it was a nuclear winter.
It absolutely was. Companies were shuttering left and right. Investors were fleeing. And it just felt like the innovation engine had stalled out.
But the data in front of us today tells a completely different story. It looks like the thaw has finally hit. We're not just seeing a recovery, we're seeing what some analysts are actually calling a biotech renaissance.
And that's the key thing.
What strikes me about this list of 20 startups we're looking at isn't just that they have cash again, it's what they're actually doing with it.
That is the whole story right there. We are not talking about marginal improvements.
Yeah.
We aren't talking about a slightly smaller pill or, a generic version of something from the 90s. The companies we're looking at today are engineering things that sounded like pure science fiction just a decade ago.
It really does feel that way. We've got AI supercomputers that are acting like full-blown co-scientists. We have gene editors that act like pencils instead of scissors.
And digital twins that let us test drugs without ever touching a human being. It's a fundamental philosophical shift.
A shift from what to what?
From treating biology, managing symptoms as they appear, to actually reprogramming it.
So here's the roadmap for Mo today. Whether you're an investor scouting the next unicorn or just someone who plans on inhabiting a body for the next few decades and wants to know how it works, this matters.
It really does.
We've organized this into 4 main themes. We're going to start with the brains, the AI revolution.
Then the tools, next-gen gene editing.
After that, the logistics, the FedEx problem of drug delivery, and finally, the massive market of metabolic disease.
And we have to wrap up with the business reality check. Because even with the coolest science, companies still go bust.
The wins, the losses, and the graveyards.
Yep.
Okay, let's start with the brains. Segment 1, the AI revolution. Now, AI in drug discovery has been a buzzword for 5 years. But looking at Recursion Pharmaceuticals ticker RXRX, it feels like we've moved past the hype into something real.
We absolutely have. Recursion is so interesting because they realized very early on that biological data is messy. You can't just scrape the internet for it like ChatGPT does with text. There's no clean internet of biology.
You have to generate the data yourself.
You have to. And that's the Recursion operating system for that.
Scale is just hard to wrap your head around. They are running actual wet lab experiments.
Millions of them. They have this massive automated facility in Salt Lake City. I mean, think of a factory floor. But instead of building cars, robots are taking cells, stressing them with chemicals, breaking them, and photographing them.
And they run how many a week?
Over 2 million experiments.
Yeah.
Every single week.
It's brute force biology, and they're feeding all that proprietary data into BioHive 2.
Their supercomputer built with NVIDIA. And that machine creates a map of biology. See, human scientists, we're biased. We look for what we expect to find.
You have a hypothesis, and you test it.
Exactly. The AI just looks at millions of images and finds relationships, correlations between a gene and a disease that a human would never, ever notice.
And they made that huge power play back in late 2024, merging with Exscientia. That felt like a turning point.
Oh, that was the missing piece of the puzzle.
Yeah.
Recursion was the master of the broad search screening everything to find a target.
In Exscientia.
They were the masters of precision design, actually building the chemical molecule to hit that target. Merging them created this full-stack juggernaut.
And this isn't theoretical anymore. They have REC 3964 in the pipeline for C. diff infections. And the mechanism is fascinating because it doesn't actually kill the bacteria.
No, and that's the genius of it.
Yeah.
Traditional antibiotics are like carpet bombing. They kill the bad bacteria, but they also wipe out your entire gut microbiome.
Which leaves you vulnerable.
Right. REC 3964 targets the toxin the bacteria produces. It neutralizes the weapon, not the bug. It spares your microbiome.
It's just a smarter approach. But while Recursion is doing this brute force screening, you have In Citro, led by Daphne Koller, taking a more simulated approach.
Daphne's a legend in machine learning. And In Citro is betting the farm on digital twins.
This concept blows my mind. Explain it for the listener.
Okay, so imagine you want to test a bridge design. You don't build a physical bridge and then drive a tank over it.
You build a digital model, you simulate it.
You simulate the tank. In vitro is doing that for human biology. Instead of testing a drug on a mouse, which is a terrible predictor for humans, in vitro takes human data from stem cells and uses machine learning to build a predictive model of a specific disease.
So they create a mathematical version of ALS or liver disease.
Essentially, yes. A digital version of the sickness. And this lets them run simulations to distinguish correlation from causation. That is the classic trap in biology. In vitro's models are designed to tease that apart.
And Big Pharma's buying in. They've got deals with Eli Lilly and BMS.
It's validating the tech. But then you have the new heavyweight that just walked into the ring with $1 billion. Xaira Therapeutics.
When you launch with $1 billion, you are just not playing the same game.
Not at all. They launched in April 2024, and they are swinging for the fences with generative AI. The key distinction here is de novo design.
Meaning from scratch.
Right. Most drug discovery is like digging through a huge bucket of keys, hoping one fits a lock. Xaira says, forget the bucket, let's measure the lock and 3D print a key from scratch.
It's incredibly high risk.
High risk, high reward. But with a Nobel laureate on the team and that kind of war chest, they can afford to fail a few times.
So that's the AI landscape. It's not a tool anymore, it's a co-scientist.
Which brings us to gene editing. Once you find the target, you need the tools to fix it.
Segment 2. The tools. Everyone knows CRISPR, the molecular scissors. But reading the updates on Beam Therapeutics, it feels like scissors is an outdated analogy now.
It is. We are moving to the era of the molecular pencil. See, traditional CRISPR-Cas9 works by cutting the DNA double helix in half. It snaps the strand.
And the cell panics and tries to repair it.
Exactly. It's chaotic. The repair process can introduce random errors. It's great for breaking a gene, like smashing a light bulb to turn it off, but it's terrible for fixing a specific typo.
So what does Beam do differently?
They use base editing. They don't cut the double helix at all. They chemically convert one letter of DNA into another,
Right.
an A to a G, for example.
So they just erase and rewrite.
That's the analogy. And the safety profile is theoretically much better because you aren't shattering the genome. Look at the Beacon trial for sickle cell. The data from mid-2025 was incredible.
It was a functional cure.
Absolutely. Patients showed durable increases in fetal hemoglobin, but the kicker,
Yeah.
0 vaso-occlusive crises.
And for those who don't know, those crises are the hallmark of sickle cell. They are these excruciatingly painful events. Going to 0 is life-changing.
It's a home run. But even Beam is editing the sequence. Chroma Medicine and Chroma Bio is asking, why touch the code at all?
This is epigenetics, the software of the cell, right?
Think of DNA as the hardware, the hard drive. Epigenetics are the volume knobs that tell the cell which programs to run. A liver cell and a brain cell have the exact same DNA.
But different genes are turned up or down.
Precisely. And Chroma's tech just grabs that knob and turns it. They can silence a gene without breaking the DNA. And the critical difference here is reversibility.
You could, in theory, turn it back on.
Exactly. It's a huge safety advantage.
It feels so much less invasive. But we can't talk about editing without talking about cancer. Caribou Biosciences is tackling one of the biggest logistical headaches in oncology, CAR-RT therapy.
CAR-T has been a miracle for blood cancers, but the process is a nightmare. You have to extract a patient's T cells, ship them to a lab, engineer them, ship them back.
It takes weeks, and sometimes the patient just doesn't have weeks.
Exactly. Caribou is building off-the-shelf CAR-T universal donor cells. You could have them stored in a freezer at the hospital.
The issue has always been rejection, though, right? My immune system attacks them.
That's the hurdle. Caribou uses a hybrid CRISPR tech called sheet-HRDNA to essentially put a disguise on these donor cells. They knock out the markers that scream foreign invader.
So the patient's immune system just ignores them.
While the cells go and attack the cancer. If they pull that off, cancer therapy becomes a product you buy, not a bespoke service you wait weeks for.
But all of this, I mean, the AI design, the gene editing, it all fails if you can't get the drug to the right place.
Which is segment 3, the delivery challenge, the FedEx problem.
Right. You can have the cure for a muscle disease, but if it ends up in the liver, it's useless.
Yes.
Or worse, toxic.
And the body's a fortress. It's designed to keep things out.
So let's look at Dyne Therapeutics. They're focused on muscle diseases like dystrophy, which are notoriously hard to penetrate.
It's dense tissue. Dyne's solution is clever. They use what they call the FORCE platform. They realize that muscle cells need a lot of iron, so they're covered in transferrin receptors, basically doorways for iron.
So Dyne attaches their drug to an antibody that acts like a key to that specific door.
It's a Trojan horse strategy. The antibody latches on, the cell opens up to let the iron in, and the drug slips inside.
And from a business perspective, it's fascinating. You have Avidity Biosciences with similar tech getting acquired for $12 billion. Dyne is sitting there with a much smaller valuation.
It shows how valuable delivery tech is. If you solve delivery, you solve the disease. Look at SpliceBio. They're taking on the hardest target of all, the brain.
The blood-brain barrier keeps out 99% of drugs.
So SpliceBio decided to stop fighting it and just go around it. They deliver gene therapies directly into the spinal fluid.
Intrathecal delivery, that sounds intense.
It is invasive. It's essentially a spinal tap in reverse. But they are treating these rare, fatal pediatric diseases like GN1 gangliosidosis. There is no other option.
It's a lifeline.
It is. And then you have the liver experts, Arrowhead Pharmaceuticals. They work with RNAi.
The mute button for genes.
Right. Arrowhead solved liver delivery years ago with their TRiM platform. What's exciting now is they're moving outside the liver, targeting the lungs, the central nervous system.
The big selling point I saw in their data is the durability.
That is the game changer. We are not talking about a daily pill. We're talking about an injection maybe once a quarter or twice a year.
Imagine managing a chronic genetic condition with 2 appointments a year. That changes the psychology of being sick.
Absolutely. Speaking of which, let's talk about the topic that has completely dominated the cultural zeitgeist, metabolic disease.
The Ozempic era.
Right. Segment 4. We know the GLP-1s work, but they are injections. Yeah. Nobody loves needles.
So Structure Therapeutics is trying to change the form factor.
Their holy grail is the oral pill, GSBR 1290.
Why is this so hard? Why can't we just mash Ozempic into a pill?
Because Ozempic is a peptide, a large biological molecule. If you swallow it, your stomach acid destroys it like a piece of steak. You just digest it.
So Structure is designing a small molecule.
Synthetic chemistry that mimics the effect, but is tough enough to survive the stomach. And the implications for access are huge. Manufacturing sterile injectable pens is expensive and complicated.
It's a supply chain nightmare. We saw the shortages.
But pressing pills, that's cheap. If you want to treat global obesity, hundreds of millions of people, you need a pill. It's the only way the math works.
But it's not just about weight, is it? It's about the damage to the organs, specifically the liver. Xaira Therapeutics is focused on MAH.
Metabolic dysfunction-associated steatohepatitis, a terrible acronym for a terrible disease where the liver gets so fatty and inflamed it starts to scar.
That scarring is fibrosis, and it leads to cirrhosis.
And liver failure. Until recently, the only cure was a transplant. But Xaira's Harmony study, that data was stunning.
What did it show?
74% of patients saw an improvement in their fibrosis. The drug didn't just stop the damage, it started to reverse the scarring.
That's regenerative medicine in action. You are turning back the clock on liver damage.
It really is.
So we've covered the AI brains, the molecular pencils, the delivery trucks. It sounds like a victory lap, but we promised a reality check.
We did. And this is segment 5, because for every one of these successes, there are a dozen autopsies.
Let's start with the big win, just to set the bar. Karuna Therapeutics.
The success story of the decade for neuroscience. For 50 years, we treated schizophrenia the same way, blocked dopamine. The side effects were awful. Karuna took a gamble on a completely different pathway.
And that gamble paid out $14 billion when BMS acquired them in March 2024.
It proved that neuroscience isn't a graveyard. But then you have Spotlight Therapeutics.
Shuttered in February 2025.
They were trying to do gene editing in vivo injecting the editor directly into the body. And they just couldn't solve the delivery hurdles before the funding ran dry.
It's a reminder that possible doesn't mean easy. And then there's Xaira. I remember when they were the darlings of synthetic biology.
A classic cautionary tale. Cool tech, no market. They were using biology to create high-tech films for folding phones.
Yeah.
Amazing science. But the electronics market moves too fast. They couldn't scale manufacturing. They just burned through cash and imploded.
And sitting somewhere in the middle is Ginkgo Bioworks, the organism company.
They wanted to be the app store for biology. We build the platform, you build the apps. But the problem with being a platform for biotechs is that when biotechs run out of money,
you run out of revenue.
Exactly.
Yeah.
They've had to restructure massively just to survive. It shows that the business model is just as important as the science.
So pulling all these threads together, we're sitting here in January 2026. What's the big picture takeaway?
The word is convergence. 5 years ago, biology was biology. Chemistry was chemistry. Computing was computing. Now, Look at Recursion. They are a tech company. Look at Structure. They're doing hardcore chemistry to solve a biological problem. Dyne is engineering molecular vehicles. The silos are gone.
It's all blending into one discipline, bioengineering.
And for you, the listener, the so what is profound. We are seeing the definition of treatable expand in real time. We are moving from managing symptoms to fixing the root cause, a genetic typo, a scarred liver, a chemical imbalance in the brain.
It makes me think back to in vitro. If we can build a digital twin of our biology, are we approaching a future where we cure diseases in a computer before they ever even manifest in our bodies?
That's the dream. Preventative maintenance for the human machine.
A fascinating thought to leave you with. Keep an eye on these 20 names as 2026 unfolds. It's going to be a wild year. Thanks for joining us on the Depth Dive.
Stay curious.